So in this unit, we're going to relate what we learned about genetics into more of the grand scale of populations and look at evolution. So, the modern theory of evolution recognizes that the main source of variation in the population lies in the differences in genes that are carried on our chromosomes. And it's the genes that determine an organism's appearance. Mutations can cause new variations, and those variations can be passed on from one generation to another. And so, we think about, for example, human populations, and if you look at historically, right, there are parts of the world where a trait might be more pronounced than another part of the world simply because they're mating within those populations and those genes are getting passed on within that population. And then genes in another population are getting passed on within that population. And each of those populations are um, exposed to different selection pressures that are different from one another, very likely, that are going to cause certain traits to be more pronounced or less pronounced, depending on what those selection pressures are. So when we look at human populations, and really any population actually, rather than testing or getting information from every individual in a particular population, we can do a random sample. So a random sample is going to test a small group of a population. And then what we do is we apply what we learned about that small group and say, well, it's likely that if that small group has a particular trait, let's say 80% of them have a particular trait, it's likely that that entire population, probably 80% of them have, or around 80% of them have that particular trait. So random sampling is a really important way to test populations where you don't have to actually test every individual. You can apply what you know from a random sample to a larger group. And one of the activities that I do in class is we look at the trait for tongue rolling. So if you can roll your tongue, sort of make it a U shape, kind of a circle shape, that's a genetic trait that's dominant. It has to do with a muscle gene that causes your tongue to be able to make that formation. And about 65% of the population have that trait. So if we were to look at our school and say, okay, we've got 2,000 students in our school, rather than check every one of those students to see if they can roll their tongue, I could send students to different classrooms and we could do a random sample. So let's go to 5 different classrooms in different parts of the school, ask the teacher if you can do a quick count of how many people can roll their tongue and how many can't. And then we come back, we get that data, we put it on the board. And from that, let's say 70% of the students at our school could roll their tongue. Well, that means probably that 70% of the students in the school can probably roll their tongue. Of course, there's got to be a lot of conditions, that we got to make sure that we're choosing and testing our variables correctly, but we could make that very, broad assumption. When we recall from Biology 20, we do need to recognize some definitions as we're moving into this unit. The first one is a population. A population consists of all of the members of a species that occupy a particular area at the same time. So, for example, the perch, which is a type of fish population in Lake Wabamun in the summer of 2019, or the dandelion population in a school field, over however many years or even just one season. The reason that a population is important is because members of a population are more likely to breed with one another than with other populations of the same species. So if we had a grizzly bear in Kananaskis and a grizzly bear in Jasper National Park, they're not likely to breed with one another. But grizzly bears in Kananaskis are likely to breed with grizzly bears in Kananaskis, and those in Jasper are more likely to breed with Jasper. But because there's that distance between them, they're not going to be mating with one another. And so if one of those grizzly bear populations has a unique trait, either from a mutation or that was passed on along to them from their parent, they're more likely to stay in those populations because they're breeding within their population. Humans are a little harder to work with because, especially, in the last 100 or so years, we've been able to travel across the world differently than what we did in our earlier existences. In our earlier existences, it was very secluded populations in terms of, and traits being carried on, right? We could have a woman move across the world and have children with a man from another part of the world with different genes, and you get more of a mixture, right? The grizzly bears aren't going to be able to do that. So the total of all of the genes in a member in a gene pool, or sorry, the total of all of the genes in all of the members of a population at one particular time is called that population's gene pool. And evolution is the change in the frequency of genes in that gene pool over a period of time. So we would expect them to change over time. And if you think about why, well, why would genes change over time? There's mutations. There's going to be, let's say, some organisms that aren't going to reproduce and pass on their traits, right? There's going to be some evolution that's always going to happen. But what we're going to do when we talk about Hardy-Weinberg, which is on the next page here, we're going to see how Hardy-Weinberg look at the idea of evolution and changes of genes in the gene pool. So since the discovery of DNA, we have a lot more information in terms of what goes on in the cells, in terms of genes and alleles. So when we're looking at populations, so this unit in particular, we're looking at the bigger range. So this isn't just, mom has brown hair, dad has light hair, and that this is my hair color. We're going to look at, where did that hair come from in that bigger, that greater population of people. And humans again are more difficult to work with. So I'm going to give an example with moths. So if you consider a population of moths, they're going to be in this big cage. There's two alleles that we're going to study here, the uppercase B and lowercase b. Uppercase B is the dark brown wing, has a dark brown wing, and that's dominant. And lowercase b is light brown wings, and that's and that's recessive. So if I have 500 moths in that cage, and 480 of them are dark brown and 20 of them are light brown. Of that 480, let's say I've done genotype testing on them, and I know that 320 of them are an uppercase B and an uppercase B. 160 of them are an uppercase and a lowercase b. So of that 480, I know how many are homozygous and how many are heterozygous, and then the remaining 20 are lowercase b, lowercase b. If all of those moths were allowed to mate, and then let's say they have 500 offspring, I take that 500 offspring and I'm going to get them out of the cage. Let's imagine I can actually do that with these moths. And I'm going to count how many of them have dark wings and light wings. Would I find that there are 480 that are dark and 20 that are light? Would I find the same proportions? Okay, and then even so, would I say of that 480, I've got the 320 and the 160? Well, in theory, according to Hardy and Weinberg, yes, they should be the same, because whatever kind of goes into the recipe comes out of the recipe. So if those are the alleles that we're starting with, there's no extra moth mating, every moth is mating, then we're going to expect the same proportions coming out. So when Hardy and Weinberg said that, if we think of the reality of it, we think, well, is every moth going to mate? What if the dark-winged moth is more attractive, and so more of the moths want to mate with a dark moth, or that it's easier to see, so it's easier to mate with? What if one moth escapes, or a moth comes into that cage and starts adding another allele, a different proportion of alleles to this population? Well, Hardy and Weinberg are assuming none of that happens. Hardy-Weinberg equilibrium is sort of like the ideal gas law of chemistry. So we're going to put all of these conditions on this environment, and which are pretty unrealistic. But if we can put these conditions on and say, if this, and this were to remain stable, then we would expect that there would be no change in terms of the allele frequency. But when we see that, oh, as a matter of fact, there is change in the allele frequency that happens, we say, okay, well, which one of these got violated then? There was a mutation in there. That's what changed the allele frequency. There were only, 460 dark, and there were 30 light, and then there were 10 this sort of spotted color. Where did that come from? I don't know if I did the math there right. But that's where you can see after. We're going to put these assumptions on. When the calculation doesn't work out the way you think, then we go back and say which one of these got violated. So the 5 components of Hardy-Weinberg equilibrium are very important for you to know. So the first is that the population must be large. It's sort of no different than if I was to flip a coin 10 times, I could actually get 8 tails and 2 heads. But if I was to flip a coin 100 times, I'm going to get more likely to the idea that it was going to be closer to 50-50. If I was to flip a coin 1,000,000 times, chances are I'm going to get even closer to the predicted outcome of a 50-50 split between heads and tails. So the larger the population you're working with, the closer you're going to get to achieving Hardy-Weinberg equilibrium. The other piece is that no migration can occur. So I talked about the moths. What if one of them slipped out? What if another one came in from another population and brought in that spotted trait? There can't be any migration that occurs. Every one of those moths also needs to mate as well, and they must mate randomly. That's the last point there. So every one of those moths, if you have a moth that doesn't mate, well, that moth might as well just not even be in the cage, because they're all contributing these alleles to the next generation. If it's not contributing, it's got to be out. The other thing is no mutations can occur, really something we don't have control over, but we're going to assume that no mutations have occurred, and no natural selection. So nothing that provides a survival advantage to one particular set of moths. And that's sort of described in the next set of points here. So if a population is small, chance is going to play a greater role in terms of causing changes in allele frequencies. If there's migration, we're talking about removing alleles from a population and adding alleles to a population. We don't want that occurring. When mutations occur, we're new alleles that are arising then now can come into that population and be passed on from one generation to the next. If natural selection occurs, we can't have anything that provides an advantage or greater reproductive success. And mating needs to be random. There can't be something that makes a mate more visible or more attractive. Okay, so when they're violated, when any of these conditions are violated, we're going to get gene pool change, which eventually might result in evolution. And we call this disrupting Hardy-Weinberg equilibrium. Okay, and most of the time, Hardy-Weinberg equilibrium is disrupted in one way or another. But by imposing these conditions, we can see how the change, or see the change happen, and then say which one of those conditions may have been violated. So if we think about even in terms of them individually, so I'm just going to pop over to the next page here, we do need to know a couple terms relating to this. So when we look at the violations of Hardy-Weinberg, that first one that has to do with the population being small can cause what we call, in an overarching term, genetic drift. So if the population is small, changes that occur by chance are going to affect a smaller population more than they would affect a larger population. So for example, if you had an allele that's found one time in every 100 organisms, let's say, and you have a population that has 500 individuals in it, that would mean that 5 of them carry the allele. So if it's a 1 in 100 chance of having this particular trait, that a population of 500, there should be 5 individuals that have this particular trait. If the population is 50,000, then 500 would carry that trait, right? It's that 1 in 100 chance. So if we imagine this to be, let's say, 2 islands, okay? So I'll put island 1 and island 2. Okay, they're about the same size in terms of their island. This is population 1. This is population 2. And our population 1, we had 5 individuals that have this particular trait in that 500. And in population 2, we had 500 of them, because there's 50,000 people in that, or individuals in that population, and they're islands. And so there's a hurricane that comes through, and it equally affects both of those islands. The chances, let's rate this a hurricane. The chances of 5, let's say, in each of those islands, 50 people die. Well, if 50 people die, if 5 of those people, of those 50, are ones that have that trait is now no longer in island 1. But if 5 or 50 of them die, and let's say all of them just happen to have that trait in population 2, you still have 450 individuals with that trait. You haven't lost the trait, or it hasn't been wiped out. So the larger the population, if any chance event like this hurricane, let's say, that comes through affects the population, you're more likely to have survivors that still can contribute that particular trait moving forward. It's like, if I had $1 million and I lost 10,000 of it to to some sort of an online scam, let's say, in my $1 million world, that doesn't seem to be much of a dent. But if I only had $30,000 and I lost $10,000, that's a lot of money in terms of, because I'm starting with a small amount in the first place, right? If I want to relate it to money, that kind of helps as an example. So we call that, as a general term, genetic drift. When we speak of it specifically, there's two general types of genetic drift we talk about. The first is the bottleneck effect. The bottleneck effect is kind of similar to when you think about a bottleneck while driving, right? So if you're going from a 3-lane highway to a 1 lane, right? Everyone's trying to merge into that middle lane. You get this bottleneck. So a bottleneck effect in terms of populations is, if you look in these bottles here, and I'm using bottles as this analogy, you have blue and yellow alleles. So the blue and yellow alleles in the parent population are on the far left. Something happens in that environment where not all those alleles can get passed on to the next generation. So the ones that are getting the ones that are going to survive are any of those alleles that are getting dropped into this bucket right here. So for whatever reason, more individuals that have blue alleles survive this particular bottleneck, whatever it is that's preventing the population from reproducing. And so if you have more blue alleles in the surviving individuals, when you look at the next generation, you can see proportionately there's actually more blue marbles in this bottle than there were at the beginning. And that's because there was more blue individuals that survived. Thinking about an example in real life, seal hunting. If you think about hunting of seals drastically reducing the population in about 1890. So let's say hunters that are hunting seals hunt, they're going to be more successful at hunting the seals that perhaps are more easily visible to the hunter. And so that, let's say it's, a little bit darker of a color or lighter of a color, whatever the case could be, then you're going to see less of that color in populations moving forward of the seals, because they were more visible to a predator, and that predator has now removed those alleles from the population. Those seals can't reproduce and pass their genes on to the next generation. The second type of genetic drift that we talk about is called the founder effect. And there's sort of two general types of founder effect. The first is if you think a few individuals from a large population leave to establish a new and small population. So think about, pioneers and settlers, right? So you might have this large group of individuals, and you have a couple individuals in that group that decide to move on to another area and start their own population. If those few that decided to leave had a particular trait that was different than that other group, or even just proportionately, they're going to be a little bit different in terms of their alleles, and then they start a new population in a different area, upon generation, that's going to be different than what their original population was that they came from. Another example, if you think about a seed that's carried by wind or a bird to a new location, where you've got this population of dandelions in a field, and you're playing soccer in the field, and you come home, but you got dandelion dust on your shoes, and you walk on your front lawn. Now you're bringing those alleles from that dandelion population into your lawn, right? And they're going to start a new population there. Another type of founder effect is called, or due to a spontaneous mutation. Sometimes that happens. You might have an organism that, if you've got a population of bears, they've got an extra, all of a sudden this one bear was born with an extra toe on its left front paw. So now it has an extra toe, and if that bear lives to survive and reproduce, and then it's going to pass on that extra toe allele to the next generation. Okay, so it's sort of like it just comes up as it's sort of founded. It just shows up. It's almost out of nowhere, it seems, but that's obviously not the case when we're talking genetics. So on the next page here, gene flow is kind of the general blanket term we use to talk about the effect of migration on Hardy-Weinberg equilibrium. And if we have gene flow, then we have migration, immigration, or emigration. And we don't want gene flow in Hardy-Weinberg equilibrium. An example of gene flow that happens in real life is prairie dogs, kind of like larger squirrels, kind of gopher-like mammals. They live in dense colonies that have just about a few dozen members, but then For much of the year, they don't allow any other members to come into their colony, but in late summer, male pups are actually permitted to enter new colonies. So come in and mate with the female prairie dogs. And then that's going to affect gene pools, because where are these male pups coming from? They're coming from an outside population. Mutations in general, mutations are randomly occurring events that are alter in the inheritable genetic material of an individual, and mutations can be favorable or unfavorable mutations. And genetic mutations will become common if they're beneficial to the individual who possesses them. And that's because natural selection will favor things that are beneficial. Then we move into natural selection. The best example of natural selection that we talk about in biology is sickle cell anemia. So it's a recessive, autosomal recessive trait, and it affects the hemoglobin in red blood cells and thereby affects the ability for a red blood cell to bind with oxygen. The other thing that sickle cell anemia does, and many people don't know this, especially from North America, and if you do, that's awesome. But what it does is it provides resistance against malaria. And malaria is a parasitic infection that's carried by mosquitoes. And that when the mosquito bites that has a parasite on it, bites a person, it will then infect the blood and the blood cells. So malaria is really very, very, very rare and unlikely in North America. And it's because the mosquitoes don't live along to harbor that kind of a parasite. But in areas where they don't have the full cold winters like we do in Canada, and you don't have that chance for like rebirth or resurvival of mosquitoes, those mosquitoes live a long time and they have a longer life cycle. And then that gives them more chance of actually harboring parasites such as the malaria parasite. And it infects red blood cells. But if a person has sickle-shaped blood cells, the parasite can't attach to it. And so people who have sickle cell anemia, or even those who are heterozygous, they actually do still have some blood cells that are sickle-shaped, have a resistance to malaria. So having a condition like sickle cell anemia is a benefit in those areas where malaria has a high incidence, because as long as the sickle cell anemia can be treated, right, through transfusions and so on, and medications for those individuals, the sickle cell isn't going to be as detrimental as dying from malaria. And so if you have sickle cell anemia in those areas, it's a benefit. If you have sickle cell anemia here in North America, let's say, it is not a benefit, because the only sort of thing that you get from the sickle cell anemia are those related to decreased oxygen in the body. So those individuals are going to be more tired. They're going to probably have a shorter breathing rate, trying to get more oxygen in, and so on. So you can see how natural selection can favor one environment and not another. So that sickle cell anemia example is always a really good one to explain natural selection. So sickle cell anemia, we could say it's a harmful mutation if we're talking about somebody having that mutation who lives in North America where malaria is non-existent, or very low anyway. There's very few places in Canada where it could survive, or in North America. It's a beneficial mutation in countries where malaria it has a higher incidence. So let's say somewhere like, in Central America, like Belize or Guatemala. It might be a beneficial mutation because it's better to have sickle cell anemia and be protected against malaria, because malaria is more likely to kill you. So what we'll often say is harmful mutations, they can occur frequently, but they're selected against. So in that case, if there's a harmful mutation, the environment determines whether it's harmful. And most harmful mutations are selected against in that if you have that mutation and it prevents your ability to reproduce, and outside of the sickle cell example, then you're not going to pass it on anyway, because you're not reproducing, because it's, it may have killed you sooner, let's say, or preventing you from reproducing. Beneficial mutations are more rare, but when they're selected for, you're going to find those alleles more in the population over time, right? Because they actually provide a benefit. And so if you look at some data as to, how many individuals in certain areas of the world, if you looked up a map of people who have sickle cell anemia, a world map and Google Images, you would actually see way more people with sickle cell traits in areas where there's more tropical regions without those cold winters like we have. And then the last piece of Hardy-Weinberg equilibrium that needs to be considered when we're looking at those 5 ideal conditions is non-random mating. And so there can't be a trait that provides an advantage for mating or more attraction between 2 organisms. Now, we know that, again, that's not realistic, but what Hardy and Weinberg are doing are saying we're going to put these conditions on it, and then from there, by if we see what we think is evolution happening, then let's look and figure out which one of these conditions might have been broken. So there is going to be this idea of sexual selection in sort of sexual dimorphism. So if you look at different striking differences between male and female organisms, or behavioral differences between the sexes. So you look at a peacock, for example, that's a male bird, and they're very brightly colored, very, trying to attract the mates. Sometimes it's behavioral, right? Like a female fish won't drop their eggs until a male fish swims just the right way around to show that that male fish is strong enough, has, good fin movement, and is worthy of having that female fish drop her eggs so that he can fertilize them, because really it's about the prolongation of life. So that female is like, I've harvested all this energy into making these eggs. I want to make sure they have a good chance of surviving. So I need a strong mate in that regard. So sometimes it can be behavior, and sometimes it can be those physical phenotypic things as well when we talk about non-random mating.